Well, thank you all for staying for the last session, which is about 25% of renal cell carcinoma tumor types. First, I want to thank the organizers, particularly Brian and Susan and the KidneyCAN team, the volunteers who are here, who make it happen. Now, this is the third year. Shout out to Tony and Hans as well. Thank you for putting this wonderful symposium summit. I want to also send some cheers on a good note. A few days ago, we heard about the announcement of Nobel Prize in Medicine or Physiology, and congratulations and cheers to the two winners this year. One of them is a former student, not of mine, but a former student, an alumnus of my alma mater, the American University of Beirut, Ardem Pataputian, and his colleague Davis Julius, for their discoveries of neuroreceptors of touch and temperature. So this is great news for people like Tony, Ziad, who's here, and myself, to see one of our own win the Nobel Prize. This is the first time a Lebanese American wins the Nobel Prize in medicine or physiology. And this comes especially at a tough time for our country of 4 million people with a catastrophic economic meltdown. So this gives hope to all the young people to do research and science to help mankind. So cheers for that. So now we get to the business of non-clear cell RCC. I'm joined by my colleague and co-chair, Ari Hakimi from Memorial Sloan Kettering. We have 5 outstanding presentations and speakers. So it's a pleasure to start the session with a keynote from my colleague, Giannicola Genovese from MD Anderson. He's an assistant professor in the departments of geomedical oncology and genomic medicine at MD Anderson, who will speak to us about mouse models for, I like his title is variant histology rather than non-clear cell, defining those variant histology with which they're not. So mouse models for variant histologies RCC. Johnny.
Thank you. So in respect of social distancing, I'm gonna get off the podium. Okay. So let's see. So thank you for the generous introduction, and thank you for the opportunity to be here and kickstarting this discussion. Okay. So I don't have any relevant disclosure. So before getting into the business of the data of the science, I want to introduce the team. I guess Nizar, Pavlos, Tony, Ziad, Ari don't need an introduction in this audience, but I want to mention that not only the work on aggressive variants on non-clear cell tumors, but the entire renal cancer program in my lab was really spearheaded by Federica Carbone, who is now an independent investigator in Italy, and Luigi Perelli, that is still in my lab and is becoming a postdoc. And computational work was supported by the Traction platform led by Tim Heffernan and Samra Turalic from the Greek Institute. So when we started working on kidney cancer, the overarching question was, what drives the emergence of metastatic clones, of clones that have metastatic competencies in tumors that usually don't do very badly, that are relatively indolent? And I think this question is still an open question. One of the reasons is because we don't have appropriate model system that capture the complexity of human RCC, and the complexity that we appreciate at the clinical level, at the pathological level, and now also at the genomic level with all the studies that have been coming out in the last 10 years. So we basically try to bridge that technological gap by using a cross-species functional genomic approach. And basically we're leveraging the mouse as a model. So, and we're using a CRISPR-Cas9 based somatic mosaic editing approach. So really, it seems complicated, but it's actually pretty simple. So what we basically generated conditional model of renal cancer expressing the Cas9 nuclease in a tissue-specific fashion, along with a fluorescence or luminescent reporter. And then we basically transduce those animals with viral vectors, viral construct carrying packages of guide RNA targeting the genomic region of interest and the genetic drivers of RCC. So basically here, the recombinase that is provided in the mouse activates the Cas9 endonuclease, primes the reporter system, and only in the cells that are transduced by the virus activates the flip recombinase, which in turn finally activates the fluorescence reporter. So now you're able to track the fate of the cells that you've been editing with the CRISPR technology. So we've been using these technologies in the last 3 years, I would say. We basically generated about 20 different models of renal cancer, spanning the different pathological subtype, the different molecular and genetic subtype of kidney cancer. And since today we're talking about the variant histologies, I'm reporting here an example of an F2-driven tumors. And you can immediately appreciate that those tumors tend to recapitulate the pathological feature and the histomorphological features of the human disease. So now we'll be monitoring these experimental cohorts and the other experimental cohorts with other genetic alteration for about 2 years now. But we really noticed the emergence of metastatic disease, which was our original question, only in a small percentage of tumors. And this is suggestive to us that you really need something else to kickstart the metastatic process. So if you look now at the human data, the renal cancer genome, you would immediately notice that the emergence of features of aggressive disease and metastatic dissemination is usually associated with very specific genomic event. Between those, the loss of chromosome 9p and the acquisition of complex karyotype. And this is something that has been very elegantly shown by Samra Turalich and the TracerX folks, but also confirmed by other groups, including Tony and recently Bob Moser at MSK. So we basically went back to the mouse and we wanted to test this in our model system. So we performed some chromosomal engineering. Basically, we used a series of guide RNA to chop a region of chromosome 4, which is synthetic with the human 9p. And this region that is highly conserved is spanning a number of we can call them professional tumor suppressor gene involved both in cell cycle regulation and in immune regulation. Just to give you an example, MTAP, CDKN2A and B, and the interferon cluster. And by doing so, now we were able to generate very aggressive tumors that are not only locally invasive, but they also have metastatic capabilities. And in about, I would say 70% of cases, we have observed bulky metastatic disease in splanchnic organs, lungs, and I think it was about a little bit less than 10% of case brain metastasis, which is something that is not been described before. Now, from a histopathological standpoint, those tumors are poorly differentiated carcinomas with areas of sarcomatoid differentiation. And if you look at the cohort at the entire cohort, you will see that by doing this chromosomal engineering, you are actually causing a significant drop in survival, an increase in metastatic burden, and also that the pattern of metastatic dissemination, the anatomical distribution of the metastasis is very similar to what we see in the aggressive variant RCC. But to conclude, I think that the most interesting piece of data actually came from the cross-species genomic analysis. And in fact, you can immediately notice that the patterns of copy number gain and losses are highly consistent between the species, with loss of chromosome of murine chromosome 12, which is synthetic to human chromosome 14, being the most enriched event in metastatic disease. And I think this is a very good example of cross-species convergent evolution, and is telling you that this is a pretty faithful model, and that if you provide the appropriate genetic drivers, the evolutionary bottlenecks that are shaping the cancer genome are conserved between species. So with this, I'm going to conclude, I think. Okay, good. And just wanted to thank my group and mention that this work was supported by the Moonshot Program, the Koch Center, and Barbara Massi Memorial Fund. Thank you.
Thank you, Johnny. We'll take questions about Dr. Genovese's presentation after the second presenter, Dr. Richard Link.
So it's my pleasure to introduce Dr. Link. Dr. Link is a urologist, so it's near and dear to my heart, but is professor at Baylor University, where he runs the comprehensive kidney cancer program there. Dr. Link.
Thank you. Well, thanks so much for this opportunity to speak. That's a hard act to follow. That's why this is so much fun. So let's see, we're going to jump ahead here. All right, so today I'm going to touch on some of our work developing papillary renal cell carcinoma models, and I have no disclosures associated with this work. So, as a kidney surgeon, sometimes we lose track of the fact that 30% of kidney cancer patients present with advanced disease. And especially for those that have distant disease, the prognosis is still fairly poor. And this has driven the development of some incredible tools over the last several decades for treating this problem. But one of our challenges is, how do we use these tools? How do we organize them? In what order do we use them? And this can be challenging. And learning how to do that is dependent on understanding the genes and pathways involved in tumorigenesis and progression. I'm going to focus today on papillary renal cell carcinoma. It's about 20%, a little less, of the kidney cancer out there. And there's a lot of interesting reasons to study papillary. First of all, a lot of the research tools for papillary have been lacking. It's a very diverse phenotypic subtype where there are variants that are highly aggressive and those that are not so aggressive. And I think that that broad range of clinical behavior is very interesting to study from a genetic standpoint. Back when we started this project, there really were no functional models that were clinically relevant for papillary renal cell carcinoma. That's changing, but that was part of the driver. So doing functional genetics in this area was difficult. We had very few cell-based resources at that time, and generating PDXs from patients with papillary has proven to be rather difficult. So we decided to try to develop a model for papillary renal cell carcinoma. I don't have time to go through all the details, but it's a pretty simple model. It's based on the classical Sleeping Beauty transposon mutagenesis system, and it's expressed in a kidney-specific fashion, in this case using PepCK-CRE. And we've layered onto it several sensitizing mutations, either a heterozygous loss of P10 derived from the experience in Cowden syndrome, where we see an increase in papillary renal cell carcinoma, or an activating mutation of MET, which is seen in familial forms of papillary. So there are two models I'll touch on today. One is the PAP-B model. That has just got the Sleeping Beauty transposon and a heterozygous P10 loss. And then we've layered on a constitutively active version of MET onto the PAP-A model. To do that, there was no constitutively active tissue selectable version of MET at the time, so we made one using homologous recombination. This just shows that data. When we look at these mice, when activated, you see a significant increase in phosphorylated MET, and downstream effectors of MET, such as STAT3, are upregulated in that model. Those animals are viable and fertile and don't have a renal phenotype when you look at them just alone. However, when we layer on the sensitizing mutations, you see that these mice do develop tumors. In the PAP-B model on the right panel, it's a rather long period of time to develop tumors, usually average about 22 months, and they generally get one or two very large tumors at that time. In the PAP-A model that includes the MET gene, you see multifocal tumor development much earlier with an average of about 10.8 months. And if you look at tumor development on this curve, you'll see a big difference between the blue line, which is PAP-A, and the orange line, which is PAP-B. Excuse me, that's a typo, PAP-B. Dr. Michael Ibman at Baylor helped us by blindly looking at 164 specimens from these mice. We also slipped in a bunch of human tumors to try to confuse him during the process. And what you see from this data is that if you look at the PAP-A model, and the PAPI model, you see that it's very heavily skewed towards papillary in terms of the histology. And when you compare PAPA and PAPI, there's a slight difference in that the PAPA that contains the MET mutation pushes that phenotype a little bit closer towards the type 1 papillary, as you might expect based on the human experience. So I am a surgeon. I'm not a bioinformatics expert, although I'm playing one today on TV. We've had some really wonderful collaborations with real bioinformatics experts, particularly Christian Corfu at Baylor. But obviously we're interested in whether this model has any relevance to the human disease, and looked at that primarily through RNA-seq techniques. This is a principal components analysis. And it just shows that there's a grouping of the two mouse models. Those are black and red in this case, with the gray, which reflects the TCGA data for papillary renal cell carcinoma. And it separates from chromophobe and from benign tissue and clear cell. Again, when you do a hierarchical clustering of this, you see that the mouse models cluster with the human papillary from the TCGA. And if we kind of look at this in a different format, another question you might ask is, are we modeling just one variant of papillary renal cell? Or are we looking at a spectrum of variants that are more relevant to the spectrum of human disease? These are color-coded based on human versus mouse. And you can see that they distribute across that range. So we're modeling several different variants that are similar to the human disease. Again, when you look across all the TCGA, you see that the PAPA model lights up particularly papillary and clear cell renal cell carcinoma. And it's not a kidney-specific only factor in that chromophobe is far down on the list. So it seems to have some relevance to the human disease. When you look at pathways and upregulated and downregulated genes, you see that there are quite a few in common between the two models, but some that are separate. And if we compare them to the TCGA, particularly in upregulated pathways from the hallmarks, you see they're fairly similar. Less so in the case of downregulated pathways. So there's a lot to study in this area. So what's next? Well, obviously gene discovery is part of the point of this. Since we're doing transposon screening, we're well along on this process and have a list of candidate genes for both models at this point. We're in the process of doing validation for that. And then obviously we'd like to be able to identify tumor clones for testing and for further study. We've been able to derive cell lines from many of these tumors. These cells can be reintroduced back into nude mice and generate tumors that maintain their histology. So it's an interesting system for further exploitation. So we developed two clinically relevant models, I think, that are for papillary renal cell carcinoma, one MET dependent, one not MET dependent. These are highly penetrant and multifocal, particularly in the PAPA model. We've generated this MET activation model that can be useful for other systems. And I think we've confirmed a role for PTEN and certainly for MET in specifically driving the type 1 papillary subtype. Thank you very much. People to thank, particularly Dr. Idman for the pathology support, our bioinformatics expert, Dr. Corfu, and a lot of guidance and mentoring from Dr. Neil Copeland and Nancy Jenkins.
All right, we open up the floor for either presentation.
Hey, Johnny, great presentation. So you may know that Peter Kim's lab from UNC a few years back published a paper knocking out INK4A P16. They actually promote metastasis. So INK4A P16 actually locates on 9P21. So you also saw the same thing with 9P21. Do you think that is due to INK4A loss in your case?
That's a good question. I mean, I think it contributes for sure. If you remember the work from Ron DePino in pancreatic cancer and Tyler Jacks, that was in 10 years before, they showed that is actually the combination of both INK4A and ARF on the CDKN2A locus, like contributing to the more aggressive phenotype. It might be the same in renal cancer, but specifically because the guide that we design basically chops the entire area, we can discern between those. What we can do is design guides specifically on exon 1 of CDKN2A, so you just take off INK4A. So that will be like a way to address your question.
Okay, I think we're going to move it along just so that we can get to all the talks because it is a little bit late.
Just a quick question for Gianni.
Sorry.
In the corner over here.
Yes, please.
But
go ahead.
any luck in terms of engineering a clear cell histologic subtype?
Yeah, so we have several iteration of VHL with BAP1, SETD2. They tend to form tumor. They're usually, again, non-metastatic unless you knock out P53 or remove the 9P, the murin 9P locus. So it seems that you really need to mess up with the cell cycle checkpoint to kickstart that process.
Okay, our next speaker is Dr. Lisa Henske, who is a professor of medicine at Harvard and a medical oncologist at Dana-Farber and Brigham. And she'll be speaking to us about pathogenesis and therapy of chromophobe renal cell carcinoma. She won an R01 a few years ago on her work, and she is going to be presenting her work. Thank you, Lisa.
Thank you, Nizar. I'm super happy to be here. Can you hear me? Okay.
Yes.
Great. This is my very first meeting since COVID. I'm just so happy to see friends and meet new people. It's been great. So chromophobe research in Boston is definitely a team effort. We have a lot of big things coming up in Boston, actually. The Boston Marathon is on Monday, and the head of the Charles Regatta is in just about 10 days. And that sort of reflects this amazing group that we have in Boston focusing on chromophobe. It's kind of amazing that we have so many people interested. So we have a group working on single cell sequencing that includes David Braun. I'm not going to talk about that today. And then we have a talk looking at pheroptotic cell death. And all of this work that I'm doing is with my co-investigator, Carmen Priolo, who's at the Brigham and Women's. So chromophobe, as you may know, I don't have it on this slide, but the reason I'm even interested in it is because it occurs in Birt-Hogg-Debay syndrome and tuberous sclerosis. It has very unusual features, including whole chromosome losses, a high rate of mitochondrial DNA mutations, and a low number of driver mutations. And it also has a very unusual cell of origin. It's the intercalated cell in the distal nephron. And lastly, has very high levels of GSH. And what I'll tell you about today is that we think this dependence on GSH is making these cells very sensitive to ferroptosis induction, and we hope that this might be a lead toward targeted therapy. So this is a work that Carmen and I published in 2018, the first metabolic profiling of chromophobe. And among other things, you can see where the red arrows are that there are markedly high levels of GSH. And this was confirmed by David Meyerhofer's team in Germany in a paper published in Cancer Research in 2020. You can see in green here that the top metabolite that's upregulated in chromophobe relative to normal kidney is glutathione, and it's 100 times higher than normal kidney. It's really a remarkable result and confirms what we had found in that PNAS metabolite profiling using 2 different platforms, in fact. So I'm not going to tell you all of the in vitro studies that we've done, but I do want to tell you about 2 cell lines that we use and just a plea for people to try to develop more chromophobe cell lines. We really need more valid tools in this field, but we're using the UK276 that Nizar mentioned earlier that were developed by Marston Linehan, and then a cell line called Chromo-A that was developed a long time ago by a group in Germany. So ferroptosis was already beautifully introduced by May, so I'll just tell you a little bit about it. First of all, it involves lipid peroxidation that leads to a non-apoptotic cell death. Lipid peroxidation is inhibited by GPX4, so inhibitors of GPX4, such as RSL3 that May mentioned, can induce apoptotic cell death. And of course, GSH is required for this lipid deperoxidation, basically. And that can be inhibited by cystinase and by limiting the uptake of cystine and cysteine into cells via the XCT importer, which can be inhibited by drugs such as Avastin and IKE. So these drugs, Avastin and IKE, that block cysteine import cause marked decreases in intracellular GSH. These are in the UOK cells, the chromophobe model. And most of the work I'm going to be talking about was led by a very talented junior faculty member, Long Zhang, in the lab. So you can see here that the inhibitor RSL3, the inhibitor of GPX4, has more sensitivity for the chromophobe cells, the Chromo-A and UOK cells compared to 7860 cells. We see the same with inhibitors of cysteine import, IKE and Avastin, and the same for cystinase, which of course degrades cysteine. So based on these and other in vitro data, we moved forward to treat the subcutaneous UOK276 derived tumors here with IKE and found that this drug definitely inhibits the growth of these tumors. You can see the size and then also some pictures of the tumors treated with IKE. We see the same if we knock down the cysteine importer SLC7A11. We see that that also decreases the growth of the UK276 cells. And then lastly, we've used cystinase. This is in collaboration with Everett Stone and George Giorgio at the University of Texas at Austin. You can see that cystinase decreases the growth of the UK276 tumors. So this is my last slide. We believe that chromophobe tumors are hypersensitive to ferroptosis induction and that this stems from these very, very high levels of GSH that they seem to require for sustained growth. Just to, again, bring home this point that GSH is 100 times or more higher in chromophobe compared to normal kidney. We see in vitro sensitivity to all of the drugs that induce ferroptosis and in vivo sensitivity to IKE and cystinase. I just would say that this is unpublished. This is actually the first time I'm presenting this. We really would love collaborations of all types to try to move things forward in chromophobe. So thank you very much.
All right, our next speaker is Srinivas Viswanathan, sorry, from Dana-Farber, who is a MD PhD medical oncologist and a rising star in our field.
Thank you. So I'm happy to talk a little bit about some of our ongoing work in translocation kidney cancer. Disclosures. So for perspective, everything we're talking about in this session deals with a minority of the pie of kidney cancer. And translocation kidney cancer is a very small piece within that, but nonetheless, I think it's very genetically defined and has the potential to teach us a lot of important lessons about cancer biology more broadly. So the defining molecular alteration in TRCC is an activating gene fusion involving a MITF-TFE family transcription factor, usually TFE3. And clinically, TRCC compares unfavorably to other subtypes of RCC, both in the localized and metastatic disease states. And I think this is probably due to two reasons. One may be the inherent difficult biology of TRCC, but a second may be an incomplete understanding of that biology and the ability to nominate really compelling molecular targets. And I think that there have been several that are in various stages of preclinical development, but a lot remains to be done. So we've been interested in leveraging integrative genomics to get a better understanding of various facets of the biology of TRCC. And this relatively reductionist schematic shows various aspects of the biology of TRCC that I think elucidating further would be beneficial in terms of nominating new targets. So first, for example, what is the cell type within which the defining fusion in TRCC, the TFE fusion, arises? What is the precise genomic context within which that fusion arises? Are there any additional critical initiating genetic lesions that cooperate with the fusion? And once the cancer develops, what are the key downstream pathways of the fusion or in collateral pathways that can be exploited for therapeutic benefit? But one of the limitations to answering any of those questions is a comprehensive molecular understanding of the landscape of the disease. And prior studies have been limited in terms of cohort size because this is such a rare histology. One approach which we've used is to try and aggregate TRCC cases from larger genomic data sets to build a larger cohort. And I won't talk about that in detail today as CI will be presenting it tomorrow, but the short answer is that there don't seem to be a lot of other co-occurring events other than the TFE fusion itself. But all of those studies have really used either exome or panel sequencing. And it's important to keep in mind that exome sequencing, while comprehensive, is really sampling only 1% of the DNA of the cell. And so the landscape, the molecular landscape that it provides is still relatively hazy. So more recently, we've been trying to apply whole genome sequencing to better understand the genomic underpinnings and the genetic events that occur in TRCC. And this allows us to identify the precise breakpoints of the fusion, which generally occur in intronic regions, and then also the genomic context within which that fusion arises. So for example, there are certain cases in which the fusion arises in a perfectly balanced reciprocal rearrangement like is shown here. but other cases in which the fusion arises as one event in a series of more complex genetic rearrangements. And then genome-wide, looking beyond the fusion itself, there's also kind of a spectrum of genetic alterations that we've observed in TRCC, ranging from cases where you really see almost no copy number changes at all and no alterations other than the fusion itself, like the case on the left, which could be essentially mistaken for a normal sample, except for the fact that it very clearly has a TFE3 fusion detectable. And other cases in which we see larger scale copy number events, usually at the arm or chromosome level, some of which are positively or negatively selected in response to therapy. But in aggregate, I think all of this together is really showing that despite very comprehensive profiling, genetically, the key event is clearly the fusion, and in some cases it appears to be the singular event. And so that raises the question of sufficiency. And indeed, the TFE3 fusions can easily transform fibroblasts, whereas interestingly, the wild type MIT TFE genes are much less efficient at doing that. And the fusion is also a critical vulnerability of TRCC cells, whereas MIT TFE transcription factors are typically dispensable in many other cell types. So what about the transcriptional features, which presumably are downstream of the fusion in part? And so to look at this in a little more detail, we've been trying to integrate various data types, including kind of bulk RNA sequencing of TRCC tumors, single cell RNA sequencing, and sequencing of cell lines in which we make perturbations to either the fusion or cooperating factors. And I think using all of those three methodologies is important for disambiguating what are the transcriptional features of TRCC that are coming from the underlying cell of origin in which the fusion arises? What are the transcriptional features that are directly downstream of the fusion itself? And what are the transcriptional features that are being contributed by both the tumor cells and the microenvironment? And how do those differ in TRCC versus other histologies? And all of these transcriptional features are really reflections of the underlying epigenetic landscape of the cell. And so epigenetic profiling has also revealed that TRCC is really in a distinct class in terms of the landscape of enhancers. And some of those enhancers and some of the epigenetic landscape may, again, reflect the underlying cell of origin, while others are directly influenced by the function of the TFE fusion. And I think disambiguating those two facets is important for nominating what the important molecular targets are in this disease. And then finally, given the prime kind of central importance of the fusion, we've been interested in kind of understanding exactly what the important sequence features are within the fusion itself that dictate oncogenicity. And we can do this in a targeted fashion by making mutations in specific regions of the fusion. And you can see on the left that some of them impair the oncogenicity of the fusion, or a more unbiased approach like is shown on the right, where we've done subsaturation mutagenesis tiling across the entire fusion. And you can see all the dots at the bottom corresponding to specific precise residues within the fusion that, when mutated, lead to a fitness defect of TRCC cells, which I think is a prerequisite to really understanding what the critical residues are to fusion, conferring fusion oncogenicity, and ultimately targeting the fusion directly. So to conclude, TRCC is still a major unmet need, clinically aggressive. And I think that with an improved molecular landscape, improved understanding of the molecular landscape from some of the profiling approaches that I showed, in conjunction with functional approaches that really try to understand how the disease initiates and progresses, we'll be able to hopefully nominate a new host of targeted molecular targets in this disease. So I'm going to close by thanking not only the members of my lab, but everybody else across our community who's dedicated their time and expertise to study TRCC. Thanks.
Again, we'll take questions at the end of the session. In the interest of time, I'd like to present Dr. Msaouel, who will be speaking about a novel mechanism of pathogenesis of renal medullary carcinoma.
Thank you for the kind introduction. It is a pleasure to present our latest data on the pathogenesis of renal medullary carcinoma. Let's see. These are my disclosures. None are pertinent to this presentation. So the clinical associations about renal medullary carcinoma, it's a disease that was first described in 1995. It mainly afflicts individuals of African descent with sickle hemoglobinopathies. sickle cell trait, sickle cell disease, SC, anything that can make the red blood cells sickle in the medulla, as we described 2 years ago in this meeting. Median age 28 years old, 2-to-1 male to female ratio, and right-sided kidney in about 75% of cases. Again, we described the mechanism that may be causing this right-sided laterality, this predilection 2 years ago. And also, renal medullary carcinoma is always negative for SMARCB1, the tumor suppressor. And also, it's a disease that is completely refractory to the anti-VEGF TKIs that we give for other kidney cancers. And in contrast, it tends to be sensitive to platinum-based chemotherapy, but still, despite our best therapies, about 90% nowadays of patients diagnosed with renal medullary carcinoma will die within 3 years of diagnosis. Very deadly disease, so clearly we need to diagnose this earlier and potentially prevent this. So what we described 2 years ago as well is that hypoxia, and the hypoxia induced by the sickling of the red blood cells in the medulla. And that hypoxic damage appears to be the hallmark of RMC. And it is because of this and the fact that the right renal artery, as we said 2 years ago, is longer than the left artery. You get this increased resistance and reduced blood flow through the medulla that incites RMC. But what are the other risk factors? Okay, we know that if you have sickle hemoglobinopathies, you are at increased risk for RMC. Well, there is no known familial clustering. I have not heard of a single case to date of RMC occurring within families. And then there was a really nice study published recently with a haplotype-based analysis, which found no germline alleles, no alterations in any cancer predisposition genes or genes affecting kidney injury associated with RMC. So if it doesn't seem to be anything else genetic, it could be environmental. So what environmental risk factors could be interacting with the sickle hemoglobinopathies to be causing RMC? So here is what we know also about sickle cell trait. We know that it has a relationship with high intensity exercise, whereby individuals with sickle cell trait who do high intensity exercise due to the dehydration get increased red blood cell sickling, causing rhabdomyolysis. This is a well-established event. In this case, is potentially even sudden cardiac death. And so we hypothesize that this same exercise-induced hypoxia in the renal medulla of individuals with sickle cell trait will also facilitate the pathogenesis of RMC. And to test this hypothesis, we first started with models, mouse models developed by the mouse modeler extraordinaire Genovese and PhD student Melinda Sung, to evaluate the effects of exercise in mouse models of sickle cell trait. And here you can see how the mouse models recapitulate what we see in sickle cell trait versus wild type beta hemoglobin, human beta hemoglobin in mice. You can see that we have the red blood cell sickling mainly in the medulla, in the renal medulla, as we have in the humans. In addition, we took this mouse model and we fused the HIF-1 alpha oxidant-dependent degradation domain to firefly luciferase, which allows the non-invasive monitoring of HIF-1 alpha activity. And HIF-1 alpha is a marker of acute hypoxia, as opposed to HIF-2 alpha, which is a marker of chronic hypoxia. And so HIF-1 alpha activity in response to hypoxia using in vivo imaging systems. And we show here, we knew that already, you know, based on our existing literature, but we show here as well that the right renal artery is longer than the left renal artery. Again, as we discussed 2 years ago, that fact that you have the longer right renal artery increases the red blood cell sickling in the right medulla versus the left. So our mouse model would recapitulate this phenomenon. And what we did is we took that mice and had them run on treadmills, and had them run on treadmills in ways that induced high intensity exercise, which is physiologically established to be same as high intensity exercise in humans. And what we found is that indeed, both in the left kidney, but mainly in the right kidney, the high intensity exercise is associated with increased hypoxia, increased firefly luciferase. Unfortunately, the slide, the colors don't show well here. Significantly increased HIF-1 alpha activity compared to the wild type, mainly pronounced in the right kidney. In contrast, moderate intensity exercise did not do that. It actually was associated with a significant decrease in HIF-1 alpha activation in the setting of sickle cell trait. So what may be happening here is that the vasodilation of moderate intensity exercise may be counteracting the effect of the red blood cell sickling that happens. So that may actually be a good thing. So while high intensity exercise exacerbates the renal hypoxia in the setting of sickle cell trait, moderate intensity exercise mitigates this hypoxia, particularly in the setting of sickle cell trait. Now, armed with that information from our animal models, we saw what happens, in the clinical situation. And we did a retrospective chart review, a case control study, essentially, all of the patients with RMC diagnosed at MD Anderson until December 2020. And we used as controls patients with similar stage, similar age, and similar biological sex with other malignancies, stage 4, essentially almost all of RMC is stage 4, seen at the same time period at MD Anderson, same department, same time period, work led by our urology fellow at the time, Dan Shapiro. And here we show that these were well matched with significant differences in race. And we know that race is a risk factor for RMC due to the association with sickling. And here we see the significant association with sickling. Interestingly, our patients with RMC were less likely to have other comorbidities, and our patients with RMC had worse on average ECOG performance status because RMC is so deadly and so aggressive. But they were much more likely to report retrospectively using the same way as the control, higher history of exercise. And then we objectively measured the exercise activity by measuring skeletal muscle mass by cross-sectional imaging obtained prior to starting systemic therapy. This allows the quantification of skeletal muscle and subcutaneous fat surface areas. And I think that a picture here is worth 1,000 words. RMC matched. See here before therapy, RMC matched how much more skeletal muscle mass these patients had. Remember, this is an aggressive cancer, so sarcopenia goes against us. So these are more likely to have sarcopenia than the controls, and yet we see the difference, statistically significant difference as shown here as well. Then to analyze this, we had to use and populate also methods used in epidemiology. That includes, part of this toolbox is the Mendelian randomization mentioned before, essentially instrumental variables. We wrote a review on that experience to communicate the use of these tools for urologists and oncologists, so feel free to read this. And by using these techniques, we're able to explicitly represent our model, and this is our model of RMC pathogenesis, and what we set out by doing the multivariable analysis is refuted. So we tried to refute our model with a multivariable analysis, and as you can see, p-value again statistically significant. And so we found that individuals with reported history of high intensity exercise had 10 times higher odds of being diagnosed with RMC, and its increase in skeletal muscle surface area by 1 centimeter square by meter square increased the odds of being diagnosed with RMC by 4%. Again, on multivariable analysis, those are the confounders we set out, but we added others as well. You can adjust it any way you want, the signal is too strong. Then we did an additional comparison, and we hypothesized that Black individuals with sickle cell trait serving in the US military will be more likely, because of the exercise history, to develop RMC than the general population of Black individuals with sickle cell trait in the US. And to test this hypothesis, we compared it with our RMC cohort, and these are the numbers. So Black individuals similarly aged with our RMC cohort, 20 to 40 years old, 1.5% serving in the military, 11% in our RMC cohort. So a higher proportion of patients with RMC have served in the military than would be expected compared to the similarly aged Black population with trait in the United States. Then we prospectively, so additional patients, now we knew about that story of exercise, we say, okay, what's happening now? Let's gather more detailed data on exercise, not just retrospectively, the activity on 7 additional patients with RMC that we saw. And here is what we got. So as you can see here, 5 out of those 7 patients, small numbers, but still quite revealing. And we're talking about, when we say high intensity exercise, these are essentially professional athlete type of exercise. So 5 out of 7, the other 2 completely sedentary. Again, small numbers, but none of these patients reported a history of moderate intensity exercise. Now, this is a video that we published even before we had noticed that connection. This is a patient not seen at MD Anderson. This YouTube video has been seen more than 5 million times now. This patient was seen with RMC in Chicago, and the connection was that, you know, he was exercising a lot and he had rhabdomyolysis. It was an interesting case. That's why it's a YouTube hit. But now we know that there is an even bigger connection. It was the fact that he was involved in such high intensity exercise that increased that risk. And so in summary, clinical observations and animal models support the hypothesis that high but not moderate intensity exercise is a modifiable, it's the first ever modifiable risk factor for RMC among individuals with sickle cell trait. Here we define what is moderate and what is high intensity exercise. Moderate intensity exercise is very likely protective. So exercise is good. Exercise would be good even if this wasn't protective. But we have an even bigger reason now to say exercise is good. So always think of sickle cell trait and RMC in young athletic, because many of them don't know they have trait. African-American male individuals who present with hematuria and a right-sided kidney lesion. And this is our potential future work now, find better screening methods for those high-risk groups and biomarkers of kidney damage in the setting of exercise. Thank you very much for your kind attention.
Thank you, Pavlos, and thank you to the speakers. We were told that we were given 15 minutes for questions because it's such an important area and also fantastic speakers and presentations. So we'll open it for questions to all the speakers. Alex, go ahead.
I have a silly surgical question. So the renal artery issue, fascinating. So 30% of people have duplicated renal arteries. Have you looked at duplication as a risk factor, since you would expect the risk to be even double if you have duplicated?
Yeah, what a beautiful question. We did not notice that. So because we have the imaging for most of these people, that's how we measure the skeletal muscle. We did not notice any association with duplication at all. We can even look into this again, but you know, it's something that we would see and we haven't seen.
The Lebanese man.
Dr. Simon, Dr. Simon.
Thank you. Are you seeing this, especially with the more long-term model? Is there any progress in that regard? Because I think that's one thing, all of the mouse models, we still lack.
So I'll just
Comments?
summarize the question because it was cut off at the beginning. Dr. Simon was asking about mutational burden, high mutational burden in the mouse models.
Okay. You want me to go first?
Yeah, go ahead.
Okay. So at least in the somatic model, the mutational burden is less than what you see in human, and is expected because, you know, it's a high throughput approach. You see tumors in less than 1 year, so you don't have the time to accumulate mutation. What was very interesting is that the mutational signature are almost identical. So, you know, it suggested again that providing the right drivers, the evolutionary pattern are very similar between the species.
I think the thing about the difference in these models, Gianni's got kind of a sniper rifle here. He's hitting specific targets. The transposon system is kind of like a shotgun, right? We're launching these transposons around the genome. Depending on the size of the concatemer that we start with, we're getting lots and lots of hits, and they'll even do some secondary jumps. So I think we're seeing a lot more mutations.
So no, great talk, Pavlos. Is there, you put the role for HIF1, is there any role for HIF2 here? Did you look? And the second question is, you know, your strategy for the detection and screening is very good, but this is quite an aggressive disease, more than even pancreatic cancer. So how would you think of screening these patients? Probably maybe not by imaging.
Fantastic questions. And actually, literally 5 hours ago, we were talking about the distinctions we're finding between HIF1 and HIF2 in RMC. there is a difference, and stay tuned. We'll get you some additional data. It's not like clear cell. There are some distinctions. With regards to screening, you're absolutely correct. This is a very aggressive cancer, and we need to develop screening methods. But if we just start doing ultrasounds in everyone who has sickle cell trait, we'll get a lot of false positives. So by identifying risk factors like this one and potentially others, we can potentially narrow down the group. Like, for example, those that are professional athletes, but even more potentially with biomarkers, we can start testing. So there's a lot of work to be done, but hopefully as we understand more about what's causing this, we'll be able to develop such strategies.
Antihypertensives that might affect renal arterial tone in some of those patients. Do you see that as a risk factor?
It's fantastic. So we are very interested exactly in these very low cost, non-toxic interventions that can reduce the red blood cell sickling. So there are, for example, certain drinks, and we have the mouse models and we can test them, that can potentially, even in the setting of exercise, reduce the red blood cell sickling. Yeah.
Yes, this is a question about for Dr. Henske about ferroptosis. So I was intrigued by the fact that the chromophobe cell lines were more sensitive than the clear cell to your ferroptosis inducers. And you mentioned that they had higher levels of reduced glutathione. So did you measure ROS or any comparison between clear cell and chromophobe or iron content or anything that might explain? Because having more glutathione would imply a more reductive environment and more protection against ferroptosis.
Yes, we have thought about that quite a lot. We haven't extensively tried to compare to the 7860 cells, but fundamentally, we think that the mitochondrial defects, which have been recognized for a long time in chromophobe, are probably driving a very high oxidative environment, and that's leading to a requirement for very high GSH to maintain homeostasis. But we haven't extensively compared to the clear cell. We're more focused on the chromophobe.
And you could block the death with liproxatin or ferrostatin or?
Yes, I didn't show that, but the death is blocked by liproxatin and not by other inhibitors, similar to what you showed with the apoptotic inhibitor. So it does seem to be a ferroptotic cell death.
Yeah, so that's really interesting. We'll have to talk more about that. Thank you.
We would love to.
Well, I'll ask a question then for Dr. Viswanathan. I think a lot of us are, the translocation tumors are obviously rare and can be very aggressive. Anecdotally, most of the trials obviously have focused and they're included in some of the, in fact, there's a joint trial with pediatric group because of the rarity and the propensity to occur in kids. And there have been some anecdotal reports of responses to immunotherapy. One of the interesting things is, given that it's a fusion in a translocation that can be very defined, there have been some thoughts that these fusions may, in some instances, be immunogenic. So I was just wondering your thoughts on that and whether there might be a scenario where you could find the right fusion and then treat that patient properly.
Yeah, I mean, I think that that's definitely something that could be going on. Certainly, several of the fusions create predicted fusion neoantigens like in your work. And I think the responses to immunotherapy are probably higher than you would expect, given that there are no other alterations in the disease. So it's probably one contributor. I think one challenge is the diversity of fusion partners. There's like over 20 different TFE fusion partners that have been described. And so given the fact that the histology is already rare and the immunogenic fusions may require a particular fusion partner with a particular MIT TFE gene and a particular HLA background, I think that would probably be the challenge, but there probably are a subset of cases if the right background in the host and the right particular fusion that may respond to immunotherapy paradoxically.
Lisa, I have a question for you. One of the objectives of your project was to look at sickle cell sequence, RNA-seq, right? And do you have any sickle cell RNA-seq that shows that primary tumors are also responsive, like the cell lines, to ferroptosis inducers?
Yeah, so the single cell work has not really focused on the ferroptosis concepts. We've more been trying to look at the tumor immune microenvironment. But that's an interesting idea. We could look in more detail on the enzymes in the ferroptosis pathway and see how those compare in the single cell data. That's a good suggestion. Thank you.
Srinivas, question to you about the translocation. So how do you explain when you knock down the TFE3 in the translocation RCC cell lines, you reduce clonicity, you reduce chronicity without effect on the clear cell RCC cell lines?
So I think the reason for that is that there's a lot of redundancy between the MIT TFE genes. And in fact, the knockout mouse for TFE3 has no phenotype. And so probably in clear cell lines, when you knock out TFE3, there can be redundancy from the other 3 MIT TFE family members, whereas the fusion is not really wild type TFE3. It's kind of probably has some neomorphic activity that allows it to be the driver in TRCC, and so they're unable to compensate when the driver oncogene is lost. So it actually represents a very good selective target for TRCC in that sense.
Any questions from the audience? Have any questions from? Okay. Can you raise your hand? Do I see anybody asking a question? Well, Pavlos, fascinating, obviously, that provocative. I mean, obviously, it may have very, very important implications for the people, subjects with sickle cell trait. I have 2 questions that are related. First, what's the lag time between exercise? I mean, that patient who presented to the emergency room in Chicago, 2010, has been lifting weights. So what's the lag time if we know about lag time between starting exercise and development of RMC? That's one question. The other, I think, obvious, I think, source to go to, and I think you alluded to it at the end of your talk, is you have the DOD. I mean, we have here, we're in DOD here. We have the military, and of course, all the athletes, but the military has all these records of people in the military who serve the country who have sickle cell trait. Do we have any access data from the Department of Defense or armed forces to look at of all individuals, men and women who serve their country who have sickle cell trait? What's how many developed RMC?
That's a great question. So the first time actually that adverse events due to high intensity exercise in the setting of trait were ever detected were in the late 70s in the US military. So it was the DOD records that led to that first identification, and that's when they instituted checking for sickle cell trait to everybody. And then what happened, I actually learned that less than a month ago from a hematologist that was giving a presentation on sickle cell trait. Then what happened in the early 2000s, there were some issues, you know, that it would affect inclusivity and whatever, and then they stopped doing it. But in the meantime, while they were checking for sickle cell trait, the ones that were positive, they would be extra careful. They would still serve in the military, exactly the same way, but they would use a little bit different regimens to reduce the risk of red blood cell sickling. Now, at the same time, as you know, in Houston, there was an athlete with sickle cell trait that died, you know, while playing, I don't remember, was it basketball? I don't remember. And since that time now with athletes, they do that. So I think that it's important to take advantage of these very well kept and records in the DOD now to seek connections with RMC. Now, with regards to lag time and even potential for, you know, different types of exercise, now that we have seen this signal and we're prospectively evaluating and being much more careful, because, you know, as physicians, as you know, we don't usually ask a lot of questions about exercise history. But now every single patient with RMC that we're seeing, we interrogate them very, very thoroughly, exactly to understand when was it started, how many times per week, what type of exercise, et cetera, et cetera. So stay tuned. We hope in the next 1 to 2 years, potentially even in this meeting, we'll be able to give some answers to that question.
So the last one about RMC. So accepting that 2/3 of patients with RMC have strong history of high intensity exercise. So that means 1/3 don't have that history, don't give that history. What do you think is then contributing to the development of RMC? I mean, obviously, it's sickle cell trait, we know that, but what is it in that 1/3 of the patients? Is there any other modifiable risk? I mean, we know for the athletes that are okay, don't do high intensity exercise. But what about the third who don't have history, who are sedentary?
I believe there is, but I can speculate about a lot of things that can potentially induce the stress that would increase the red blood cell sickling, but I don't want to give too speculative ideas because I don't want, we don't want to, create stress to people. The high intensity exercise story, we only, set it out after we really have tried to validate this. So a lot of things could be that are playing a role in that 1/3. So stay tuned. Maybe in the coming years we might get some better ideas.
I think we'll wrap it up there. Thank you, everyone. And I would just, I want to just make a quick plug for the fact that there are several smaller institutional trials that are going on right now for non-clear cell. We have a few at our institution being run by Dr. Lee, and then there are several at Dana-Farber and other institutions, of course, all the work from MD Anderson and RMC, but it's really a big Herculean effort, and we do appreciate all the efforts by the patient supporters for us to help recruit patients to these non-clear cell trials. Thank you very much.
Thank you all.
Thank you.